Hello my dear viewers, welcome to Biology at Priyajit. In today's class we explore the mitochondrial respiratory chain and ATP synthesis, uncovering how cells convert energy from food into usable ATP, the universal energy currency of life. You will learn how electrons from NADH and FADH₂ flow through the electron transport chain located in the inner mitochondrial membrane, and how this electron flow drives proton pumping, and how a step electrochemical gradient is created across the membrane. By the end of this class, my dear students, you will have a strong conceptual grip on how mitochondria power the cell and why the respiratory chain is very much central to metabolism, and how chemiosmosis event represents one of the most elegant principles in modern biology, which is essential for UG and PG level understanding. So, what is cellular respiration? Stages of cellular respiration include glycolysis, which occurs in cytoplasm, where glucose is converted into pyruvate. Then pyruvate will be converted into acetyl coenzyme A, which happens in the mitochondrial matrix, and then this acetyl-CoA will enter into the Krebs cycle or tricarboxylic acid cycle, or TCA cycle, or citric acid cycle. Then it will produce NADH and FADH₂, and finally the mitochondrial respiratory chain or electron transport chain, which occurs in the inner mitochondrial membrane, generates ATP. So these electron transport chain or ATP, which is located in the inner mitochondrial membrane, and its function is to transfer of electrons through redox reactions to generate proton gradient. What are the general features? Electron pass through a series of complexes, that is complex 1 to complex 4, and energy released pumps H plus ions from matrix to intermembrane space. This creates an electrochemical gradient. So what is complex 1 or NADH dehydrogenase? Basically NADH donates 2 electron to complex 1, and the enzyme is the NADH dehydrogenase, which is a large protein, and this complex 1 contain mainly it has 40 polypeptide chain, but mainly it has flavin mononucleotide, which is derived from vitamin B2, and the other one is the iron sulfur complex. It is the iron sulfur center. Both FMN and FES act as prosthetic group, and this complex one pumps 4 H plus ions or 4 protons from matrix to intermembrane space, and it contributes significantly to proton gradient formation. Then complex 2, which is also known as succinate dehydrogenase, that donates electrons directly to complex 2, and electrons pass to ubiquinone. And the features of ubiquinone: this is a lipid soluble protein, and it is mobile within inner membrane. It is reduced to QH2. Then Q collects electron from NADH via complex 1 and FADH2 via complex 2. Then complex 2 does not pump protons. Hence, FADH2 produces less ATP than NADH. So this is the image showing the outer membrane. This is the cytoplasm, then outer mitochondrial membrane, then inner mitochondrial membrane, intermembrane space. And this is the inner mitochondrial membrane. So within the inner mitochondrial membrane, there is electron transport chain. So there are four components: complex 1, complex 2, complex 3, and complex 4. And the fifth one is the ATP synthase, which is also known as complex 5. Both NADH and FADH2 will be converted into NAD plus and FAD, and they will generate H plus ion, and the H plus ions will be liberated from the mitochondrial matrix into the intermembrane space through the complex 1, complex 3, and complex 4 subsequently. And coenzyme Q and cytochrome C act as the electron carrier in between the complexes. Complex 2 do not transport any H plus ion into the intermembrane space. So what does ATP synthase does? It will again pass the H plus ions from the intermembrane space to the mitochondrial matrix, where the low concentration of H plus, and it will generate ATP by with the help of the enzyme ATP synthase, which is located on the complex 5 from ATP. We'll discuss in detail. So complex 3, what it does, cytochrome, it is also known as cytochrome BC1 complex. It contains cytochrome B, cytochrome C1, iron sulfur protein and rest center, that is also an iron sulfur complex. Cytochromes contain the heme group as hemoglobin. So the iron component of the heme alternates between two oxidative states, that is Fe2 is the reduced state and the Fe3 plus is the oxidized state, and then it pumps 4H plus ions into the intermembrane space. Then it transfers electron to cytochrome C, one electron at a time. Then what does complex 4 does? Cytochrome C oxidase, it contains cytochrome A and A3, copper ions CuA and CuB. It accepts electron from cytochrome C, and then oxygen binds between iron and copper. Then oxygen is fully reduced to form water, and then it pumps 2H plus ions and maintains the proton gradient. Chemiosmotic hypothesis, it is proposed by Peter Mitchell 1961. So the key points of this hypothesis, that is electron transport releases energy through the redox reactions, and energy is used to pump H plus ions across membrane, and accumulation of H plus ion creates chemical gradient and electrical gradient. Both these electrical gradient and chemical gradient ultimately produce the electrochemical gradient, which is known as proton motive force, and this proton motive force is responsible for ATP synthesis. This PMF or proton motive force stores energy that is used for ATP synthesis. So what is proton motive force? It is the energy stored due to H plus ion concentration gradient, that is the chemical gradient, and electrical charge differences, which is also known as electrical gradient. So protons move back across again. So we have already discussed in the image, previous image. So proton move back across the membrane down through their gradient, and this movement drives the ATP synthesis. So what basically chemiosmosis tells, chemiosmosis is the movement of ions, especially protons, across the membrane, mitochondrial inner membrane, resulting in an electrochemical gradient that drives the ATP synthesis. So ATP formation by chemiosmosis, this is the electron transport chain we know. So protons flow through the ATP synthase, this is the ATP synthase, and this flow causes rotation of the F0 part. This is the F0 part, this is the membrane embodied portion, and this is the F1 part. Rotation drives the F1 head, and ultimately ADP plus inorganic phosphate will be converted into ATP, and this ATP synthesis driven by PMF is called as chemiosmosis. So its site is, in case of bacteria it is plasma membrane, and in case of mitochondria it is located in the inner mitochondrial membrane. Generation of proton motive force. Definition: PMF is the electrochemical gradient of protons across inner mitochondrial membrane. So its site, eukaryotes, in case of eukaryotes it is located in the inner mitochondrial membrane, and in case of prokaryotes it is located in the plasma membrane. So proton pumping by ETC, that is complex 1 transport 4H plus ion, complex 3 also transports 4H plus ion, and complex 4 transports 2H plus. Complex 2, there is no proton pumping. So what is the structure of F0 F1 ATP synthase? It is also known as F-type ATPase, as it is found within mitochondria. So it is known also as complex 5. It is located in the inner mitochondrial membrane, and its function is to synthesize ATP using proton motive force. So it has two portions: F0 portion, which is membrane bounded structure. This is the membrane bounded structure, or F0, and this is the F1, which is the matrix side portion. So F0 portion acts as proton channel. This is the channel made up of the C subunit and C subunit. So subunits A, B and C ring is there. C act as the ring structure, they will form the channel, and A subunit is associated with the C subunit, and B subunit is attached with the A subunit, and it is located in the matrix side. B subunit is located in the matrix side. Both A and C is found in the mitochondrial inner membrane. So the F1 portion, that is the matrix side portion, there is a catalytic domain. Subunits contain 3 alpha, 3 beta, 1 gamma subunit. This is the green colored gamma, it is the rotatory subunit, and then epsilon is there, and there is a delta subunit. Beta subunit is the main catalytic site, and gamma subunits rotate inside the alpha 3 beta 3. So binding change hypothesis of ATP synthesis, and this concept was proposed by Paul D. Boyer. The main concept is the ATP synthesis occurs via rotational catalysis. Central gamma subunits rotate inside alpha beta subunits. So what happens? Alpha that is ATP synthesis, as the asymmetrical gamma subunit rotates, it forces of the three beta subunits in the F1 head through three distinct conformational states, that is open state, loose state and the tight state. Open state, the subunit has the low affinity for nucleotide and allowing newly synthesized ATP to be released and fresh ADP and inorganic phosphate to enter the site. Loose or L state, upon further rotation, the site closes slightly and loosely, trapping the ADP and PI, and the tight state or T state, in the final rotation stage, the subunit undergoes a major shape change, forcing ADP and PI together to spontaneously form ATP. This bond formation is driven by the physical deformation of the protein rather than direct input of metabolic energy, and so each full 360-degree rotation of the gamma subunit results in the synthesis and release of 3 ATP molecules, one per beta subunit. So the mechanism include the proton flow rotates gamma subunit, and rotation converts O to L and L to T state conversion, and then ATP is formed in T state. The energy source is the proton flow through the F0. Mechanism of ATP synthesis by means of binding change hypothesis. So proton flow rotates gamma subunit, and conformational change in the beta subunits. There is three conformational change. L state binds ADP and PI. T state, ATP is formed, and open state where ATP is released. So one full 360-degree rotation produces 3 ATP molecules. So to conclude, the mitochondrial respiratory chain and ATP synthesis represent one of the most elegant and efficient energy converting systems in biology. The orderly transfer of electrons through complexes 1 to 4 not only drives redox reactions but also establishes a powerful proton gradient across the inner mitochondrial membrane. This gradient, rather than the electrons themselves, becomes the true source of energy for ATP formation. By understanding this process, we gain insight into how cells power every biological activity, from muscle contraction and nerve impulse transmission to growth, repair and metabolism. The mitochondrial respiratory chain thus stands as a cornerstone of cellular bioenergetics, demonstrating how fundamental chemical principles has transformed into life-sustaining energy through precise and highly regulated biological mechanisms. Thank you.